Hello, I am Karen Cichy, a dry bean geneticist with USDA ARS at Michigan State University. A major focus of my lab's research is to improve end-use quality traits in dry beans through plant breeding. Today, PhD student Weijia Wang will speak about her research methods used to evaluate seed coat quality and canning quality in beans.
Hello everyone, today I would like to talk about the research projects that we have been working on in improving the consumer quality of dry beans. First, I want to talk about mechanical damage and seed coat cracking. Dry beans are considered as fragile in terms of its susceptibility to mechanical damage. The injury can happen in the process from harvesting to post-harvesting handling, and the injury can be visible cracking on seed coat or non-visible internal damage. The morphological structure of most seeds determines greatly their susceptibility to mechanical damage. As you can see from this diagram, injury to any part of these indicated parts of a seed will result in a defective, slow-growing seedling. For beans, the embryonic axis, where the root and shoot develop from, is positioned toward one side of the seed and protected only by a thin, fragile seed coat, which makes them very vulnerable to mechanical damage. During harvest, when the environment is very dry, the bean pods dry down too much and will result in low moisture level of the seeds. Research has found that seeds with moisture content below 12% are much more vulnerable to damage than seeds with a moisture content of 12% to 18%. When beans are handled at the local elevator, seed coat damages can be caused through dropping beans onto floors of store silos, or running through cleaning equipment or conveyors at high speed. Mechanical damage can have significant impact on the germination rate and the emergence of bean seeds. It can cause bud head seedlings, as shown in Figure 1. Seeds with mechanical damages will also result in unattractive canning products, which is a big consumer quality concern. In our lab, we do a seed coat staining test to measure the mechanical damage on our beans. I took a video to show you how this test was done. 100 seeds were randomly selected from each sample, and they were soaked in iodine solution for 5 minutes. After 5 minutes, the seeds are dried and sorted into 5 different groups. The 5 groups are Group 1, no visible seed coat damage. Group 2, 1 or 2 minor cracks. Group 3, several minor cracks or 1 major crack. Group 4, more than 1 major crack. And Group 5, split seed. And the seed numbers are counted for each category group. As we know, at local elevators, less than 10% of checked seeds is considered acceptable for a load of dry seeds, or 20% for soaked seeds. These tables show the seed coat cracking rate of the genotypes we have tested. There are commercial varieties and our breeding lines. Belt thresh means they were hand harvested and threshed with a belt thresher, which is very gentle and causes very little mechanical damage. Combine thresh means the beans were threshed through combine. As you can see, the tested kidney beans showed a very good range of susceptibility to mechanical damage, but the black beans didn't show that much variability. We want to further explore the genetic variability for mechanical resistance in our beans, especially kidney beans, and here is a video by Dr. Karen Cichy.
In order to develop bean varieties with reduced susceptibility to mechanical damage, it is important to understand genetic variability for this trait and to identify potential parental sources that can be used in breeding. So with that goal in mind, 66 diverse kidney beans were planted at the Montcalm Research Farm this season. They included 21 dark red kidney, 36 light red kidney, and 9 white kidney. Our evaluation plan at harvest is to pool entire plants and thresh the plants when the seeds reach a low moisture of about 12%. And two different threshing methods will be used. One is a gentle belt thresher to prevent damage, and the other is a Hagie-Plot combine set to a high cylinder speed that will induce damage. These seeds will then be evaluated for seed coat check using the iodine staining method. Here is a clip of what the beans look like in the field as of August 15th.
Canning quality is another important trait we are working with. I would like to show you the method we use to test for canning quality of our beans. First, the seeds are manually cleaned and the moisture content is measured. Then a certain amount of subsample is weighed and placed in a mesh bag. The seeds are then soaked and blanched, or only blanched without soaking depending on the seed type. The blanched seeds are transferred to cans with hot brine added to them. Cans are sealed with an automatic sealing system and then cooked in a retort. For post-canning evaluation, we usually do a sensory evaluation followed by other quality traits measurement. First, cans are matched with labeled paper tray, then a group of trained panelists evaluates each sample for the appearance with a score of 1 to 5, 1 be the worst and 5 be the best. With black beans, we also do score for its color. After that, samples are rinsed and weighed. A high quality image is taken with our machine vision system, which is a black box with an stable illumination source and a digital camera that is connected to a computer. We also measure texture as the maximum force to cut through 100 grams of subsample, and we measure color with the colorimeter. Here I have a video to show you how the texture analyzer and the colorimeter work. We have recently started to work with pouch processing. The goal is to process beans using shorter time duration for improved physical and sensory quality while ensuring food safety. This is what the retortable flexible pouch in our study looks like. For research purpose, thermocouple is attached to the pouch so that we can measure the internal temperature during the retort process. The advantages of pouches include they provide the shelf stability of metal cans, they provide consumer convenient ready-to-eat products. Also, they have lower carbon footprint due to lower energy requirement during food processing and transportation. Here is our method of small-scale pouch processing. First, beans are prepared following the small-scale canning protocol and placed in pouches. Then pouches are sealed with a vacuum sealer. We need to repeat step 1 and 2 for each sample. Then pouches are placed in special designed metal trays and loaded to the retort. Thermocouples on the pouches are attached to external sensors so that we can monitor the internal temperature and decide when to call a stop for the retort process and ensure the food safety. Finally, the quality evaluation is also conducted just like what we do for the cans. These photos show you how we used to do the post-canning sensory evaluation. Beans are laid on the bench and our group members come and score them for appearance. But this is how we do the sensory evaluation now. Okay, so I'm gonna show you the first sample for today. With our machine vision system, I share the computer screen with our group members through a Zoom meeting. Everyone sees the samples one by one and score them like we usually do.
Okay, cool. So you may start to score this one. This is sample number 1.
Last but not the least, we have made an infographic to tell the farm to fork story of beans. This is designed by me and illustrated by Pia Garcia, a graphic designer we work with. It consists of an infographic and a set of stickers of the characters. The target audience of this infographic are kids aged 0 to 12. The idea is to educate them about the process that our beans go through from farm to our table. We believe that kids can influence their parents in what to purchase for food. Hopefully, they will find this infographic very interesting and want to eat more beans.
Thank you for your attention. This research is funded by a USDA OREI grant. And we'd also like to give a special thanks to Everbest Organic and Saddleberg Farm for their support with this project, as well as the CS Mott Sustainable Agriculture Fellowship received by Wei Wang. Thank you.